now turn the call over to Reshma.
Thanks, Susie. Good morning, all, and thank you for joining us on short notice. Vertex's differentiated R&D strategy continues to progress in new disease areas, and we are pleased to announce results this morning from our Phase 2 proof of concept study with the selective NaV1.8 inhibitor suzetrigine in patients with painful lumbosacral radiculopathy, or LSR. Let's jump right into it with slide 3. The study met its primary endpoint, with suzetrigine achieving a clinically meaningful and statistically significant within-group reduction from baseline of 2.02 points in the weekly average of daily leg pain intensity measured on the NPRS scale at week 12 of treatment. The consistency of suzetrigine's within-group treatment effect is remarkable, with a clinically meaningful reduction in pain across all studies done to date. However, the study also saw similar reductions in the NPRS in the placebo reference arm. The placebo effect in this study was a 1.98 point reduction from baseline to week 12. In short, the treatment response curve of suzetrigine did not separate from placebo, as I'll detail shortly. Importantly, suzetrigine's safety profile continues to be excellent. Similar to the findings in our acute pain Phase 3 study, the adverse event rate in the suzetrigine arm was lower than the adverse event rate in the placebo group. Let me share 2 additional key points on this slide. Our interpretation of this study and our hypothesis emerging from this trial, informed by post-hoc analyses we conducted, is that a high placebo response in this study led to a lack of separation of the suzetrigine and placebo response curves. Importantly, we believe we can innovate in pain clinical trial design to better control the placebo effect, and in so doing, succeed in pivotal development with suzetrigine. As such, our next steps are to, 1, take all the learnings from this Phase 2 trial and innovate on study design to better control the placebo effect. 2, prepare for regulatory interactions. And 3, secure FDA's agreement on an optimized Phase 3 clinical trial design and requirements for expanding the PNP, or peripheral neuropathic pain indication, beyond diabetic peripheral neuropathy. Pending regulatory agreement, we plan to advance suzetrigine into pivotal development. To frame the challenge and the opportunity more fully, recall that peripheral neuropathic pain is a collection of painful conditions unified by the same underlying pathophysiology of nerve impairment. There are approximately 10 million patients treated for PNP each year in the U.S., representing a multi-billion-dollar market today, despite the fact that essentially all prescriptions are generic. Nearly 20% of these patients have DPN, where we are currently enrolling a Phase 3 program, and about 40% of these patients have LSR. These more than 4 million LSR patients have a significant unmet medical need. The currently available treatment options are associated with high rates of discontinuation, inconsistent dosing, polypharmacy, and off-label use. Unlike in DPN, where studies have been conducted for decades, study design, drug treatment effect, placebo treatment effect are well understood, and multiple drugs have secured regulatory approval. In LSR, no drug has been specifically approved for this indication. There are very few Phase 2 randomized control studies and no Phase 3 trials evaluating a systemic treatment. This leaves many gaps in understanding, including what constitutes a meaningful between-group reduction in LSR pain. On the backdrop of these challenges with LSR, we see opportunity. To be clear, to be successful in LSR, we need to innovate in both discovering NaV1.8 and 1.7 inhibitors, as we have been doing, and in clinical trial design to control the placebo treatment, as we see from this study. We look forward to doing so and bringing the promise of suzetrigine and our portfolio of NaV1.7 and 1.8 inhibitors alone or in combination for this difficult-to-treat condition. Given the lack of studies and standards in LSR, we set out to learn more and run a phase 2 study in this condition with 3 goals in mind. 1, gauge the magnitude of the treatment effect of suzetrigine in LSR. 2, understand the magnitude of the placebo effect in LSR. And 3, add to our knowledge of the 12-week safety profile of suzetrigine. Our aim was to gather this information via the phase 2 study, learn, and then use those learnings to inform our next steps for suzetrigine in LSR. I'll come back to this point. But before we go there, let's go to slide 6, which shows the phase 2 study design. The evaluated dose of suzetrigine was 69 milligrams once a day and included a placebo reference arm. Patients were randomized to receive either suzetrigine or placebo following a 7-day run-in period to establish their baseline pain score on the NPRS. The primary endpoint was the within-group change from baseline in the weekly average pain intensity on NPRS at week 12. We also assessed other efficacy endpoints as well as the safety and tolerability of suzetrigine. Patients on background stable over-the-counter doses of ibuprofen or naproxen were allowed to continue taking these doses, and acetaminophen was allowed as needed throughout the study. Lastly, the placebo arm was included as a reference group. No direct comparison between suzetrigine and placebo was planned, and the study was not designed nor powered for between-group comparisons. Slide 7 details the baseline characteristics and demographics, which were generally balanced across parameters between the two groups. Just over 40% of patients in each arm were on background NSAIDs and continued taking it throughout the trial. The weekly average of the NPRS was around 6. The results are on slide 8. For the primary endpoint of within-group change from baseline in the weekly average of the pain intensity on the NPRS at week 12, suzetrigine showed a reduction of 2.02 points. This is both statistically significant and clinically meaningful. The placebo reference arm showed a similar within-group reduction in the NPRS with a mean change from baseline of minus 1.98 at 12 weeks. Slide 9 summarizes safety and tolerability, which adds to the growing body of evidence supporting suzetrigine's favorable safety profile. Suzetrigine was generally well tolerated with 12 weeks of treatment. The rate of suzetrigine-related AEs was 22.9%. For context, the rate of placebo-related AEs was 32.4%. The majority of AEs were mild or moderate. There were no serious AEs related to suzetrigine, and there were no AEs that led to treatment discontinuation in the suzetrigine arm. On slide 10, we plot the time course of pain relief in the suzetrigine and placebo arms on one graph. Both the suzetrigine arm and the placebo arm showed similar reductions in the NPRS over time. The magnitude of the suzetrigine response from baseline to 12 weeks was statistically significant. It was clinically meaningful. It was consistent with clinical studies of Vertex NaV1.8 inhibitors in other neuropathic pain clinical studies. And it was also consistent with the magnitude of pain reduction with medicines approved for neuropathic pain. Yet, despite these findings, the suzetrigine and placebo arms did not separate, which was unexpected. To better understand these results, we further interrogated the data. The placebo response in this study was high. Moreover, we found high site-to-site variability in the placebo response, with some sites having high and some sites having low placebo treatment effects. Putting this all together, our hypothesis is that the high placebo treatment effect observed in this study, driven by some sites, led to a lack of separation of the suzetrigine and placebo curves in the primary analysis. Moving to slide 11. To evaluate this hypothesis, we performed post hoc analyses. Let me describe what we did, and then I'll describe the results shown on the slide. As mentioned, when we looked at the NPRS data by site we observed that there were sites with higher placebo response curves, defined as pain reduction of more than 2 points, and lower placebo response curves, defined as pain reduction less than 2 points. We segregated the sites into these two groups. Of note, approximately 40% of sites were lower placebo response sites. We then analyzed the primary and other endpoints at these lower placebo sites. What did we find when we did these analyses? In the lower placebo response sites, as shown on this slide, we found that the suzetrigine treatment effect was similar to the suzetrigine treatment effect in the primary or overall analysis. And there was a separation of the suzetrigine arm and the placebo arm. This separation of the NPRS score in the suzetrigine and placebo arms in this analysis is in line with expectations. In other words, here, given the suzetrigine within group performance of approximately a 2-point reduction, the suzetrigine NPRS pain reduction curve did indeed separate from the placebo NPRS curve. I want to emphasize that this is a post-hoc analysis, and as such comes with all of the caveats of such analyses and needs to be considered hypothesis generating, not conclusive. I put weight in this analysis because, 1, we had sufficient number of sites to evaluate the SUS and placebo effect in the low placebo sites, as 40% of the sites were such lower placebo sites. 2, we performed additional analyses that support the robustness of this effect. 3, the suzetrigine treatment effect is consistent in the lower placebo site analysis and the overall or primary analysis. And the suzetrigine response in this study is consistent with the treatment effect seen with other Vertex NaV1.8 neuropathic pain clinical trials. 4, it is also consistent with the treatment effect of other approved medicines in neuropathic pain. And 5, high placebo effect and site-to-site variability are known challenges in this space. Slide 12. In summary, the primary results from this phase 2 study in LSR demonstrates suzetrigine has a consistent and clinically meaningful 2-point within group treatment reduction in pain and is well tolerated. The adverse event rate is 22.9% in the suzetrigine arm and 32.4% in the placebo arm. Taken together, including the post-hoc analysis, we see the potential of suzetrigine to offer effective pain relief, strong safety, and an attractive overall profile for patients suffering from LSR. This is our belief. We obviously have yet to demonstrate this potential. To do so, to demonstrate this potential, we have to innovate in clinical trial design to better control the placebo effect, gain agreements with FDA on trial design and requirements to expand the suzetrigine indication beyond DPN, and show that suzetrigine is superior to placebo in a large, well-designed, and controlled phase 3 trial. This is exactly what we aim to do. Moving to slide 13. Vertex is all in on pain. This slide shows the breadth and depth of our programs. We've discussed our plans in LSR at length. Let me close with some highlights in the pain portfolio outside of LSR. We are excitedly awaiting potential approval for suzetrigine in moderate to severe acute pain with the PDUFA date of January 30th, 2025. Acute pain impacts 80 million Americans and is a multi-billion-dollar market opportunity despite being almost entirely generic. We are advancing additional molecules through clinical development, including VX-993 in 2 phase 2 studies, 1 in acute pain, 1 in DPN, as well as a phase 1 study with the IV formulation. And we continue to make progress in preclinical development with our NaV1.7 pain signal inhibitor program that may be used alone or in combination with NaV1.8 inhibitors. With that, we'll be happy to take your questions. I'll turn it over to you, Susie.
You ready, Drew?
Yes, we will now begin the question and answer session. To ask a question, you may press star then one on your telephone keypad. If you're using a speakerphone, please pick up your handset before pressing the keys. If at any time your question has been addressed and you would like to withdraw your question, please press star then two. At this time, we will pause momentarily to assemble our roster. The first question comes from Jessica Fye with JP Morgan. Please go ahead.
Great. Good morning, guys. Thanks for taking my question. My question is really about what's the right way to think about how you design and power a phase 3 trial, and what kind of effect size you would want to, size and power your trial for, to the extent that we can't really use the phase 2 to tease that out per se. And then kind of related to that, if there is the ability to identify sites that might have a lower placebo response, I imagine you're going to need to go to a lot more sites in phase 3 than in phase 2, i.e. not just be able to kind of identify these phase 2 sites where the placebo response is lower. So you talked about kind of innovative ways to manage that. Can you outline what some of those strategies are? Thank you.
Hey, Jess, this is Reshma. Really excellent questions. I'm going to give you a broad framework to think about this, because what we're in the process of doing is completing our interrogation of the data set to understand every last bit of information that we can. And then, of course, we have to go to the agency and confirm that the innovative study design that we want to pursue is acceptable to them for the indication we seek. But let me give you some broad strokes of what we're doing. So bottom line is, sometimes when you do a phase 2 study, it teaches you exactly what to do in phase 3. And that's oftentimes the case. Sometimes what you do in phase 2 and the results you get teaches you that you need to do something different. You need to innovate further in phase 3. That's what we've learned from this trial. And so this trial has been exceptionally helpful, because remember, the universe of studies in this LSR condition are small. And even in the small universe, there are unfortunate complexities, like studies are of short duration. They are not necessarily done in the patient population that we seek to work on, that is to say pure radiculopathy, amongst other complexities. So first, let me tackle your question around sites. It's pretty clear to me that we need to do some of the, let's call it, usual work that we do when you work on any kind of subjective measure like pain, like a limited number of sites, really good training of these sites, and sites really training their patients for how to respond to the questionnaires around their pain in a consistent way over time. So we will not, in all likelihood, be going to more sites, but rather going to a circumscribed number of sites. That might mean it takes a little bit longer to enroll, and that's an acceptable trade-off. With regard to study design, let me give you some examples. One thing that is done in studies with subjective endpoints is a placebo run-in. And what that does is it excludes patients who have a pronounced placebo effect when you run everyone in. You run whoever you screen, you run both groups in on placebo. You then exclude patients with a profound placebo response. Another thing that's done is enrichment. That's to say, you run patients in on the study drug, and you only include patients who have a response, i.e., if you're not going to ever feel pain relief, you're not included in that design. Some people combine placebo run-in with enrichment. You can do withdrawals as another technique, withdrawal of the study drug, withdrawal of placebo, to test whether the pain that is, a placebo response is equal when you remove the study drug. Those are a couple of examples of what can be done. There are others. And as I said in my prepared remarks, if you want to have a leadership position in pain and you want to have medicines across the pain spectrum, which is what we want to do in acute pain, oral, IV, as well as in neuropathic pain, we're going to have to innovate in bringing these medicines forward from discovery research, but we're also going to have to innovate in managing the placebo response. And I believe that we can do it. Now, of course, we have to show that, and we're looking forward to doing that in phase 3. I hope that helps.
Thanks.
The next question comes from Salveen Richter with Goldman Sachs. Please go ahead.
Thank you. Good morning. You touched on this a little bit, but could you just speak to what was different about the sites with the low placebo effect? And while, clearly there is a placebo effect, can you also expand on your confidence here that you enrolled LSR patients, just given the heterogeneity with that population? Thank you.
Yep. Morning, Salveen. I feel high confidence from the data that we have interrogated that we did, in fact, enroll patients with lumbosacral radiculopathy. You can see in the baseline characteristics, everyone had a dermatomal pain distribution, which is what we would expect to see. That I think is correct. With regard to sites and what's different about these sites, these are all analyses that will require us to think more deeply about. So I don't want you to think that this is certainly what we're going to do, but I'll give you one example. The sites that had lower placebo response rates had higher baseline NSAID use. That is obviously important information that we're going to take as we design this phase 3 study.
Thank you.
Yeah, sure thing.
The next question comes from David Reisinger with Leerink Partners. Please go ahead.
You there, Dave?
Excuse me, Mr. Reisinger, your line is open. Okay, we'll go to the next question from Jeff Meacham with Citi. Please go ahead.
Great. Thank you. And thanks for all the details, Reshma. I wanted to ask you a few things. The first is in this phase 2, what strategies did you guys use to try to mitigate the placebo response going into it? And the second thing is, beyond some of the innovations in phase 3, is there a way to take advantage of the novel mechanism or something in a phase 3 that could sort of convey to the FDA, look, this is an opioid alternative, for example, maybe looking at, at sort of unmet need and sort of the opioid segment of that population. I'm just trying to think of maybe even more creative ways to add, to the study beyond just, pain scores and looking beyond the placebo washing kind of a type of thing. Thank you.
Yeah, you bet about the details, Jeff. I'm happy to share 100% on what you say about the fact that this is a non-opioid and the magnitude of the treatment effect. And look, what I'm about to say to you is cross-study comparisons, and you have to take this with a grain of salt. But when you look at opioids that are used in, it's not exactly LSR, but an opioid, I think it's a Nucynta, was studied in a clinical trial of chronic low back pain with radiculopathy. So let's call it similar. The reduction in pain, the within group reduction in pain in that opioid was a 2 on this NPRS score. In that study, the placebo response was better controlled. So 100% that there is a lot of innovation to be had and lots of ways in which by clinical trial design, by endpoints, by statistical methods, we have to consider, work through, and gain agreement with the agency for how we can best conduct the phase 3 study. You asked about what did we do. Let's call it conventional approaches. We used all the conventional approaches to manage the placebo effect here. Small number of sites, training of the sites training the patients, really being very attentive to the inclusion criteria. All those traditional things were done here.
Okay, thank you.
You bet.
The next question comes from Michael Yee with Jefferies. Please go ahead.
Great, thanks. Good morning. Maybe just a two-part question on the subgroup analysis of the 40% of patients that you showed on slide 11. Do you think that is a solid representation of what you think the clinical magnitude of efficacy is for this drug ultimately if you were to run this?
Moving to slide 11. To evaluate this hypothesis, we performed post hoc analyses. Let me describe what we did, and then I'll describe the results shown on the slide. As mentioned, when we looked at the NPRS data by site, we observed that there were sites with higher placebo response curves, defined as pain reduction of more than 2 points, and lower placebo response curves, defined as pain reduction less than 2 points. We segregated the sites into these two groups. Of note, approximately 40% of sites were lower placebo response sites. We then analyzed the primary and other endpoints at these lower placebo sites. What did we find when we did these analyses? In the lower placebo response sites, as shown on this slide, we found that the suzetrigine treatment effect was similar to the suzetrigine treatment effect in the primary or overall analysis. And there was a separation of the suzetrigine arm and the placebo arm. This separation of the NPRS score in the suzetrigine and placebo arms in this analysis is in line with expectations. In other words, here, given the suzetrigine within group performance of approximately a 2-point reduction, the suzetrigine NPRS pain reduction curve did indeed separate from the placebo NPRS curve. I want to emphasize that this is a post hoc analysis, and as such comes with all of the caveats of such analyses and needs to be considered hypothesis generating, not conclusive. I put weight in this analysis because, 1, we had sufficient number of sites to evaluate the SUS and placebo effect in the low placebo sites, as 40% of the sites were such lower placebo sites. 2, we performed additional analyses that support the robustness of this effect. 3, the suzetrigine treatment effect is consistent in the lower placebo site analysis and the overall or primary analysis. And the suzetrigine response in this study is consistent with the treatment effect seen with other Vertex NaV1.8 neuropathic pain clinical trials. 4, it is also consistent with the treatment effect of other approved medicines in neuropathic pain. And 5, high placebo effect and site-to-site variability are known challenges in this space. Slide 12. In summary, the primary results from this phase 2 study in LSR demonstrates suzetrigine has a consistent and clinically meaningful 2-point within group treatment reduction in pain and is well tolerated. The adverse event rate is 22.9% in the suzetrigine arm and 32.4% in the placebo arm. Taken together, including the post hoc analysis, we see the potential of suzetrigine to offer effective pain relief, strong safety, and an attractive overall profile for patients suffering from LSR. This is our belief. We obviously have yet to demonstrate this potential. To do so, to demonstrate this potential, We have to innovate in clinical trial design to better control the placebo effect, gain agreements with FDA on trial design and requirements to expand the suzetrigine indication beyond DPN, and show that suzetrigine is superior to placebo in a large, well-designed, and controlled phase 3 trial. This is exactly what we aim to do. Moving to slide 13. Vertex is all in on pain. This slide shows the breadth and depth of our programs. We've discussed our plans in LSR at length. Let me close with some highlights in the pain portfolio outside of LSR. We are excitedly awaiting potential approval for suzetrigine in moderate to severe acute pain with the PDUFA date of January 30th, 2025. Acute pain impacts 80 million Americans and is a multi-billion-dollar market opportunity despite being almost entirely generic. We are advancing additional molecules through clinical development. Sacral radiculopathy, you can see in the baseline characteristics, everyone analysis, where there was separation of the curve, where the suzetrigine treatment effect was basically the same, about a 2, but we had better placebo effect, let's call it more in line with a couple of things that were different. For example, one thing that was different is the use of NSAIDs.
Post hoc analysis. Thank you so much.
Yeah, but it was clearly separated in the post hoc analysis. It was clearly not separated in the primary analysis.
Okay, that's very helpful. Thank you.
Would expect, innovate, and plan to get down to in our phase 3 trial. And I just want to be clear, we haven't done it yet. We have to show that, and that's what we are intending to do.
The next question comes from Evan Seigerman with BMO Capital. Please go ahead.
Hi, all. Thank you so much for taking my questions, and thank you for all the details today. Can you expand on whether there was any observed sleep benefit from the suzetrigine or placebo treatment? And if so, maybe quantify the magnitude of the benefit. I know that was a key secondary endpoint.
Yes. Evan, on the primary analysis, where there wasn't separation of the curves, there wasn't separation of any of the secondary endpoints, including sleep benefit. In the post hoc analysis, where there was separation of the curve, where the suzetrigine treatment effect was basically the same, about a 2, but we had better placebo effect, let's call it more in line with the broader neuropathic pain condition. The DCIS, that's the sleep scale, also showed commensurate improvement. I don't have the numbers on me, but it was clearly separated in the post hoc analysis. It was clearly not separated in the primary analysis.
Okay, that's very helpful. Thank you.
Yeah, you bet.
The next question comes from William Pickering with Bernstein. Please go ahead.
Hi, good morning. Thank you for taking my question. For the post hoc analysis, I was wondering if you could comment on the two arms. Were they well balanced in terms of baseline pain severity, and just any kind of broader comments on the robustness of that post hoc analysis? Thank you so much.
Yeah, you bet. Yes, we looked at the balance between these two groups in the post hoc analysis. One thing that we benefit from is about 40% of the sites were the lower placebo treatment effect sites, so we had enough sites to look at. And generally speaking, they were balanced, these two groups. As I said, there were a couple of things that were different. For example, one thing that was different is the use of NSAIDs. There was more NSAID use in the lower placebo group, but generally speaking, they were balanced.
The next question comes from Phil Nadeau with TD Cowen. Please go ahead.
Good morning. Thanks for taking our questions. Two from us. So I guess first on the post hoc analysis, mathematically, it seems like the only way you can get the results in slide 11, given what happened in slide 10, is if in the other 60% of sites, placebo beat suzetrigine by approximately 1 point. So can you describe the data from the other 60% of the sites? Was that difference because placebo had a -3 improvement in pain score, or did suzetrigine perform worse in those other sites than it did in the 40% of sites? That's question one. And then question two, why is the next step of phase 3 here? It seems like you're going to test innovations, different hypotheses on how to control the placebo effect. Isn't there a faster and perhaps cheaper way to do that through a phase 2 trial rather than moving into a long and expensive pivotal? Thanks.
Yeah. So Phil, you ask a very interesting and good question around the statistics. And obviously when you have an overall response where the two groups don't separate and then you do a post hoc analysis where one group separates from the other, the overall effect still has to be true. But no, it's not the effect in the other group, the higher placebo group, is basically a small difference. It's like, I don't know, 0.2 or something like that favoring placebo. But it's not a 1 point difference. And what it basically tells you is that this is not a finding by chance. And of course, we did additional statistical analyses to understand any bias we may have introduced by doing these analyses, and to understand whether this was a finding by chance. And we're satisfied that this is a good analysis. Still, it's post hoc, and it's hypothesis generating, not conclusive. Regarding your question about phase 2, this is an excellent question. I think the reason the field is plagued by not having made progress in this condition is because we haven't done large, well-controlled studies and really examined with innovative clinical designs what we can do to manage the placebo effect. Whether you call it phase 2 or you call it phase 3, we need to do a study versus placebo that's large and well-controlled that demonstrates statistically significant differences between the two groups. When you do that, you are doing a phase 3 study. I'm looking forward to doing it because I'd like to have that study then be represented in the label and broaden the label in peripheral neuropathic pain beyond DPN.
Perfect. Thanks for taking our questions.
Yeah.
The next question comes from Lisa Bako with Evercore ISI. Please go ahead.
Hi, thanks for taking my question. I hear everything you're saying. I wanted to understand sort of more about the regulatory strategy in the event that you do go down this path and, for suzetrigine doesn't show, statistical separation from placebo and LSR. What's the broader strategy for expanding the label into neuropathic pain? I know you need more than one study, and I know you have the other study going on, so just curious how you're thinking about that. And I know also, the guidelines are kind of on hold to some extent or have been withdrawn, so curious about any FDA interactions you've had on that. Thank you so much.
Yeah, you bet, Lisa. So let me go back a few steps. In neuropathic pain, we already have our program up and running for DPN. So this is all about how we expand beyond DPN. You also know that there are many conditions in peripheral neuropathic pain in that umbrella: DPN, LSR, herpetic neuralgia, small fiber neuropathy, et cetera, et cetera. Our preferred path to expand the label is to go through DPN and LSR and have those two, because in combination that represents more than 50% of the PNP indication, have those two conditions and those studies then lead to a broad PNP label. There are, of course, other ways to get there. Another way to get there would be, for example, 2 DPN studies and then studies in individual neuropathic pain conditions. In the old guidelines, the agency had particular expectations about numbers of studies and how many indications, and they would go one by one. But I think the agency has also said that they are looking to evolve from that and perhaps do a few indications, and that could lead to a broader indication. So I think that my preferred path has not changed. I would still prefer to go via DPN and PN and LSR to PNP, but there are other ways.
Okay, and then just a final question for me. Can you comment at all on discontinuations and how that was handled in the study, and then any use of rescue meds? Thanks.
Yeah. The study had high maintenance of their subjects that were enrolled. It was more than 90% in both arms. For patients with rescue therapy, over-the-counter acetaminophen was allowed in both arms, and there was high use, as is in all of these studies, in both arms of over-the-counter Tylenol acetaminophen. It's like 80% or so in both arms used PRN, that's to say, as needed acetaminophen. No differences on that.
Great. Thank you very much.
Drew, we'll take 2 more questions, please.
Yes. The next question comes from Ellie Merle with UBS. Please go ahead.
Hey guys, thanks for taking the question. Could you just elaborate a little bit more on these other PNP indications and potential strategies? So specifically, what other PNP indications do you think suzetrigine might be best suited for, and your latest thinking on development plans there? And I guess, do you have any plans to start anything here in the near or medium term? And if so, what would that look like? Thanks.
Yeah, my sentiment around PNP hasn't changed. This is underlying biology. Pain signaling, all pain signaling goes through NaV 1.7, which sets off the action potential, initiates the action potential, and NaV 1.8, which propagates the action potential. From everything we know and just foundational basic biology that has been studied over the last many decades, that's the case for all pain, not just neuropathic pain. Therefore, my impression, my perspective on the best way to get to the full peripheral neuropathic pain indication is by way of DPN and LSR, as I said, because when you put those two together, you're at more than 50% of the patients who have these PNP conditions. As I mentioned, there's certainly other ways to do it. You could do individual studies in each of these conditions, and we ourselves have done studies, for example, in small fiber neuropathy with the predecessor molecule VX-150. That one was also a positive study. The within-group treatment effect of VX-150 was about a 2. The placebo treatment effect in that study was about a 1. So there are definitely other ways to do it, but my preferred path is still to go via DPN and PNP. Those are the of DPN and LSR to get to PNP, and that's part of the regulatory conversations we need to have to gain their agreement on that.
Great, thanks.
Yep.
Last question, Drew?
Yes. The last question will come from Mohit Bansal with Wells Fargo. Please go ahead.
Great. Thank you very much for squeezing me in. Just wanted to understand the clinical significance of 1-point reduction, how meaningful that is. And it seems like, post-hoc analysis on site selection and number of sites which had low placebo response kind of creates a lot of risk. So just wanted to understand, isn't it a very risky path to take on with a phase 3 trial given the variability in LSR indication? Thank you.
Yeah, thanks for the question, Mohit. The question you ask around what is reasonable to expect for a between-group difference is truly an excellent question, and the field does not have an answer. The way the field defines clinical meaningfulness is a within-group change from baseline. And while there's no specific measure of clinical meaningfulness in LSR, broadly speaking, in the pain area, that is 2 points, within group 2 points. That's exactly what suzetrigine achieved. And as I said, it's just hard to do the analysis in LSR because there aren't many studies. But if you, broadly speaking, look at peripheral neuropathic pain, the average of all studies is around a 2. So I feel very good about the suzetrigine within group treatment effect. So what do you do with the between group? What you do is you power it for statistical significance because there isn't a agreed upon criteria for that. And again, if you look at, no one's ever taken a study to phase 3 in LSR, so there's nothing to look at there. There's no approved medicine specifically for LSR, so nothing to look there. So if you look broadly speaking at peripheral neuropathic pain, which is kind of dominated by DPN and chronic low back pain with radiculopathy, though that's kind of the study set that you'll see. All of the positive studies have a treatment effect of about a 2. Sometimes it's a 1.8, sometimes it's a 2.2, but it's about a 2. And the placebo response is something south of 1.3. There is one exception to the rule that I laid out, these numbers. It's one antidepressant study where the treatment effect was closer to a 3, but the placebo effect was closer to a 2. And so the difference between them is back down, and the achievement of statistical significance was based on 1. So there isn't an agreed upon clinically meaningful bar for between group. It's based on statistical significance. I hope that helps.
Got it. Thank you.
The conference has now concluded. Thank you for attending today's presentation.